So we're going to continue our discussion now on the single-stranded RNA viruses that have a plus strand, so meaning a sense strand. We just previously mentioned the enteroviruses such as poliovirus and Coxsackie virus. Also, the rhinoviruses are in this category, causing a common cold, and hepatitis A virus. Now continuing here on the next slide. Very important also in this category are the Togaviruses, where we're headed for the same group of viruses as the coronaviruses. Now here in the Togavirudae, we have the alpha virus as an example. These are about 30 viral species that are transmitted by arthropods, including the Western equine encephalitis and the Eastern equine encephalitis viruses that are usually abbreviated as WEE or EEE. Then we have the rubevirus that includes rubella, and the flaviviridae, the flaviviruses such as yellow fever, dengue fever, West Nile encephalitis virus, and then also the hepatitis C virus. And now here our famous one that everybody knows about now, the coronaviridae, most famously the coronavirus that's going around right now, which is a novel coronavirus that was named COVID-19. Also, coronaviruses, there are several that cause just a common cold. Then there's SARS and MERS, which were both coronaviruses. So here is this coronavirus that's going around, COVID-19, a beta coronavirus. There are various pictures online. I'm sure you've seen it. There's no escaping from this virus right now. It's the reason why we're doing this lecture here online. Take a look at it. You have in the nucleic acid core, you have RNA and the end protein. Then there's an envelope and there are spike proteins. Looks pretty nice actually. It's a pretty looking name, pretty sounding name too. But not so pretty what it can do to people. Here are a couple more images. One is a diagram here. All the same thing. It's very related to the SARS-CoV-2 virus, and the whole virus is enveloped spherical, about 120 nanometers in diameter. The RNA genome is associated with the end protein to form the nucleocapsid. And here's a little bit on the genetics. You can see it's highly related to the SARS virus from 2003. I don't want you to really study this, but just give you an idea. The only thing that's different from the SARS 2003 is basically this little red area right here, so not much. For whatever it's worth, here's the COVID-19 genome. And then here a little bit more on the types of proteins that I'm sure some scientists are studying, hopefully trying to figure out to make a vaccine or maybe antibodies that will work. So the viral replication cycle of this virus follows the typical pattern of attachment to the host cell, and then endocytosis of the virus into the host cell. We have a fusion of the virus membrane with the endosomal membrane, and then synthesis and proteolytic cleavage of the replicase polyprotein. Then the cells start to become viral factories, making just more viral RNA, in this case, the plus strand RNA. And then in the end, we're going to have synthesis and assembly of the viral particles. Once the viruses have been assembled, they will be budding from the host cells and will be released by exocytosis. Sort of a little bit here on the ecology of the virus. Humans are actually just an occasional host for this particular beta coronavirus. It's pretty common in pigs, mouse, rat, and bat. The reservoirs for the SARS coronavirus are bats, and for MERS, the Middle Eastern respiratory syndrome coronavirus is camels and bats. Primary site of infection is the respiratory tract. can also cause some gastrointestinal problems, sometimes even neurological problems, but it's rather rare. The bovine coronavirus causes mostly gastrointestinal problems, but for humans, it's usually the respiratory tract that becomes infected. Here are some of the cell receptors that the coronavirus interacts with. That's probably not so interesting for you guys, but maybe you want to take a look at that. And here a little bit more on the COVID-19 ecology. Geography, as you know, worldwide, so this thing has been going around. The latest cases, I was just looking that up, and it's staggering right now. As of the time of this recording, we have in the United States alone 123,774 cases. Just yesterday, there were almost 20,000 new ones. Worldwide, we are looking at 664,192 cases. And by the time you guys are listening to this recording, I'm sure it will be much higher. Almost 31,000 deaths, and that is bound to go up. Yeah, you can see here some of the associated diseases that are caused by these types of coronaviruses. So SARS, of course, stands for severe acute respiratory syndrome. MERS was the Middle Eastern version that was going around about 8 to 10 years ago. And SARS-CoV-2, novel coronavirus, that one causes pneumonia and acute respiratory distress syndrome. That's why people report that they can't breathe. As far as the transmission is concerned, people talk about a zoonosis here. That means it jumps from animals to people, to humans. And that is not usual, but coronaviruses are actually pretty notorious for being able to do that. When somebody says there's a respiratory transmission, that means somebody's coughing or sneezing on you, and these respiratory droplets, they land on your respiratory epithelium, and then that's how you get infected. Fomites means that it's an inanimate object, such as a doorknob, a telephone that somebody was grabbing and then you're touching it, or any other inanimate object. Somebody was touching it that was infected, and then you touching it and you put your fingers in your face, and that's how you get infected. So transmission from the zoonosis would be from animals to humans, and then human to human respiratory via respiratory droplets, and then fomites, inanimate objects that somebody touched that was infected. So the question remains, what can we do about treating people that are infected with the coronavirus that have severe symptoms? And well, hopefully there will be a vaccine soon. Vaccination is the best treatment, or prevention, I should say, for any viral disease. And I'm sure somebody's working on the vaccine, hopefully. So hopefully as soon as possible, we will be able to have a vaccination available. Until then, antiviral drugs, there's just not that much we have on the shelf. There are a couple that are sort of experimental right now for coronavirus infections, and they would include retrovirus and protease inhibitors. Now, the coronavirus is not a retrovirus, and so to give somebody a drug that would work for HIV infection, that may or may not work so well. People are also talking about the anti-malaria drug, hydroxychloroquine, that seems to work maybe in some people. The mechanism is not quite clear how that would work, though. And then there's a fusion inhibitor that is also experimental for, in general, for viruses. If you are preventing fusion, then just like protease inhibitors or other enzymes that the virus needs, if you inhibit them, that you might slow down the viral replication. So moving on now to other RNA viruses, and there we have the rhabdoviridae. Those are single-stranded RNA viruses that have the minus strand, so meaning the antisense strand, as their genetic material. The one that comes to mind here is the Lissa virus that causes rabies, and there are other in this category that cause numerous animal diseases. Of course, the rabies virus, we have that one in our pathogen groups. Then we have realviridae. Those are double-stranded RNA viruses, non-enveloped. And here we have the reovirus that causes respiratory problems. And then rhabdoviruses, those are probably the largest category of viruses that will give you sort of your stomach flu. From mild to pretty severe. It's usually not long-lasting, but it can be pretty, causing you to be feeling pretty bad for a day or two. Next are RNA viruses that use DNA. So they will use, they will start out with single-stranded RNA and then produce DNA. In order to do that, in order to go back from RNA backwards toward DNA, we need to use an enzyme that's known as reverse transcriptase. Now remember from what we said earlier that the central dogma of molecular biology means that you go from DNA to RNA via transcription. Now, if you go from RNA to DNA, you're going backwards. That means we're doing reverse transcription. And these viruses come usually packaged with that enzyme, reverse transcriptase, to go back from the RNA that they have to DNA. So the viral DNA, once they have it, reverse transcribed, it integrates into the host chromosome as a provirus. And here there's some good examples. The retroviridi, for example, the lentivirus that causes HIV, the human immunodeficiency virus, and then we have various oncoviruses in this category as well. So here is the multiplication cycle of the retroviridae. And so in the retroviridae, we have two very troublesome contenders here of viruses, and that will be the human immunodeficiency virus, HIV, and then also the flu viruses are in the same category. So let's take a look at the viral replication cycle here. So the typical attachment and then uncoding of the virus. And so the retrovirus enters by fusion and attachment to the membrane of the host cell. And then we have the uncoding step right here. Then here the viral, first we get the viral RNA in, and then we have the viral enzymes, the reverse transcriptase, that then will make DNA from the RNA template. The DNA, the viral DNA, now what's now DNA, will be then incorporated into the host cell genome, and from there, the host cell will be directed to make more viral particles. That includes the viral RNA, all the viral proteins, as you can see right here. And so some of the important medicines that are used against HIV infection are reverse transcriptase inhibitors, logically. So they inhibit this step from RNA to DNA. And if you can inhibit this step, of course, then that will inhibit the viral replication cycle. Also protease inhibitors, because these viral proteins that are then necessary to form the coat for the new viral particles, of course, they need to be cut to the right size. Just like any other coat that you would make, you would have to cut the fabric and make it fit. And so if you are inhibiting the enzymes that will cut the coat to make it fit for the virus, then you would inhibit viral replication, of course. So if the virus does its thing, then it will produce more RNA and all the other things that it needs to assemble to a new full viral particle. And here we have the viruses are assembled, and now they're budding off here. They typically take this here, they have spike proteins, they're enveloped, and once they're budding off, then they go on and infect more cells.